Yeah. So I'll switch to English as well, because I think there are people that are preferably hearing it in English. Um, and that's okay. I can easily present in English. And so if you have questions either in Dutch or English, I can understand both, so that's not a problem. So a little bit about myself. I'm Xavier. I studied technical medicine at the University of Twente, already quite a long while ago. And in that period, I also was a member of Piranha, mostly the diving part of Piranha, although I also played in the water hockey and a little bit of swimming here and there. And have in that period, I also became a diving instructor and taught quite a bit of courses within Piranha, and some of the old guys that are probably maybe still around. I might have taught them their open water course. After that, I moved to Amsterdam, where I worked at the Hyperbaric chamber in the academic medical center, and that's where I also joined SDVA. So that's why there are members of both clubs joining this video lecture as well. And during, well, already during high school, but surely during when I studied technical medicine, I realized that I have a passion for diving, and also quite interested in diving physiology and diving medicine. So already during my studies, I tried to do as much as possible of my electives with something to do with diving medicine. So one of my internships was at the Navy in Den Helder, and my master thesis was in the Department of Hyperbaric Medicine, looking at the effects of gas embolisms in the brain in pigs, and how treatment would help that inside the hyperbaric chamber. So after that, I continued on in that department, and since then I tried to focus my career on diving medicine, and that's why now 3 years ago I moved to New Zealand, where I am working as a researcher in the Department of Anesthesiology, which seems a bit odd, but it's mainly due to my supervisor, Professor Simon Mitchell, who is a world expert in diving medicine. And together with him and a second supervisor, Professor Jamie Slay, I'm conducting research on the effects of gas narcosis in diving. And with that topic, it quite fits quite well in the Department of Anesthesiology. And this research is also work that would hopefully lead towards a PhD somewhere, well, hopefully this year. I'm at the end of my 3 years research, which is the normal period here for a PhD. And then I hope to submit in the next 2 months. So after that, the oral defense is there, and then hopefully a diploma somewhere later on this year. So enough about me. We'll talk about gas narcosis. So first of all, we'll talk about a bit of predisposition, what makes people sensitive to gas narcosis, the signs and symptoms of gas narcosis, how to treat it. And after that, I will focus a bit more time on the working mechanism. And that's mainly due to the very limited amount of information, and even wrong information, that is in most diving textbooks and educational materials. If you do have questions, please ask them in the chat box, and there will be opportunities during the presentation to have moments to answer them. So this is the knowledge that most divers get to learn when they do one or multiple diving courses on inert gas narcosis. Signs and symptoms are similar to alcohol intoxication. They increase with depth, and if we ascend, the symptoms disappear. if you are at depth, it takes a little while, in the order of a couple of minutes, for the symptoms to really onset. And that's due to the nitrogen that needs to dissolve into your body and then into your brain before it's able to cause the effects of gas narcosis. And as with alcohol, the differences are quite distinct between persons, but also within a person. So one day you might feel perfectly fine at a certain depth, while the next day you go to the same depth and you might feel the effects of gas narcosis a lot more. And that also means that between divers, those symptoms or the amount of symptoms that they experience can be quite different. And partially this is due to all kind of environmental factors that make you more susceptible. And that's again similar to alcohol, where well, probably most of you can relate to that. Certain days you drink one beer and you feel kind of, whoa, this was quite a effect, where other days you are able to drink multiple beers and you're all fine, and you can keep on drinking without feeling the effects of alcohol that much. That's also one of the things that we know about inert gas narcosis, that although you're not capable of you're not getting less severe symptoms with multiple exposures, but it does show that when you dive regularly to greater depth, you're able to cope with the signs and symptoms of gas narcosis. And that's mainly due to, you know, how your body will react, and you know that you will be capable of reacting properly, as long as you make sure that the things that you want to do underwater are known to you. And that's also one of the risks with gas narcosis, that once things go pear-shaped and you have to do skills that you're not used to doing, and for most divers that means doing rescue attempts, is something you're not overly familiar with, and every situation will be different, requiring different decisions and different skills to apply. And that's where a small silly mistake can create a spiraling down into a calamity. So that's why gas narcosis is highly involved in incidents, especially at greater depth. So what are the signs and symptoms? This is a table that comes from the US Navy that they created quite a while ago, and it gives a general overview of the effects of gas narcosis. And most of you will probably be familiar with, well, the top two rows, maybe the third row, but after that, I hope that most of you nowadays understand that breathing air at those depths is not a good idea. And that's also because diving courses and diving training has evolved over the years, where let's say in the 90s, it was quite common to go to 70 meters on air to do a wreck penetration. Nowadays, we would frown upon that. Although there are still divers doing that, and especially here in New Zealand, the use of helium is quite limited. There are quite a lot of divers that still go to great depth on air. 50 meters is quite commonly done here, where in Europe that is more and more frowned upon to do. So the signs and symptoms already start at shallow depth. Some divers might experience it or feel it somewhere around 20 meters, where they feel that they're not completely in control anymore. They feel that their body is slightly off. There's a mild euphoria, and some tasks, and especially tasks that you're not mastering, don't have complete control over, they might be a little bit impaired. At 30 meters and beyond, you will see an increase of that effect. So you'll make more small mistakes, you're slower, and especially making decisions can be quite challenging, and especially making the right decision. Towards 50 meters, we'll see effects of laughter, of when you make mistakes, you tend to laugh them away. It's kind of, oh yeah, I made a mistake, and carry on with doing that. And as long as the mistake doesn't have severe consequences, that's all good. But you can imagine if that's a mistake on the basis of how much gas do I need and which direction do I need to go into, or do I need to swim against the current or with the current, you can quickly understand that can create severe consequences. And if you go deeper, 70 to 90 meters, we see great severe effects like hallucination, severe impairment, at some point manual dexterity of your fine motor control is impaired, to the point where you're completely hallucinated and even unconsciousness. So most divers nowadays understand that those greater depths diving air is not a good idea. And over the years, we see that the acceptance level of narcosis is moving shallower and shallower. Where when I started diving 20 years ago, 50 meters was still kind of considered the maximum. And in that time, the more commercial organizations like PADI, SSI, and the like started pushing that up towards 40 meters. And nowadays you even see some organizations arguing that 30 meters is the maximum for breathing air, and below that you should consider using helium in the mixture. So it is a slowly moving thing of what is accepted as a level of narcosis. But all these signs and symptoms require you, they're hard to see from the outside. So they do require a lot of self-recognition in order to realize that you're under the influence of narcosis. And there have been a couple of studies that showed that, again, similar to alcohol, the more narked you are, the harder it is for you to make a good assessment on your level of narcosis. And we all know that friend that had a couple of drinks too many and then says, I'm perfectly fine, capable of driving home, which in the end is probably not a good idea. So that's the symptom recognition is especially hard because narcosis does influence that. So it is relying on the recognition by you to your body as well, and that can be simple things if you have a discussion in the water with hand signals, that you're able to see that your body is not able to react in a way that you normally experience that body to react. That does require that you know your body quite well in order to understand that his behavior at that depth is normal. So a body that is instantly at the dive site where you put together because you're diving in the dive school and on holiday, you probably won't be familiar with the normal behavior of that diver underwater, and hence signs and symptoms recognition might be quite hard. The good part is that if you either do an ascent yourself or an assisted ascent with your buddy, you'll realize that once you get to the shallows, the symptoms disappear quickly. The only thing that doesn't disappear is the memory loss. So at depth, nitrogen narcosis, similar to anesthetics, have a memory loss effect where you don't remember what happened at depth. So you can have an argument after the dive saying, well, you were completely narked, you weren't able to do this or this, and the diver might just not remember. And that is due to the gas narcosis itself as well. But it can lead to hefty discussions after the dive. So to make it a little bit more interactive, some of you might be familiar with this graph, the Meyer-Overton correlation. So in the chat box, please type whatever you know about it and how does it relate to diving. Come on, people. How does Meyer-Overton relate to diving and gas narcosis in particular? Okay. Okay, that means I'll do a little bit more more explanation. As most of you, at least that replied, haven't seen it before. So the Meyer-Overton rule is something that is both Meyer and Overton at the change of this around 1900 did some experiments where they used all kind of anesthetic gases and saw how much they dissolve into olive oil, and put that on a graph where they compared it to their potency as an anesthetic gas. So how much of that gas is needed to create unconsciousness. And they saw that there was a nice linear relationship between the two of them. And that's what you see in this graph. In this graph, there are a lot of gases that are used in anesthetics. Some of them are still used, some of them are not that much used anymore, but you can see that their solubility in olive oil, which relates to the solubility in all kind of lipid structures in the body, they are, there is a nice relationship with their potency. So in diving, this is still used, but then focused on the top part where you see nitrogen, where we have a short list of the gases that are more common in diving: nitrogen, oxygen, and carbon dioxide. And we see that that based on those numbers, divers are making decisions on how narcotic a certain gas is. However, over the years of after these two guys published their research, because this doesn't explain how the gases are narcotic at a cellular level. All kind of theories have been proposed, and in order for me to explain those, we'll have to do a little bit of explanation of how neurons work and how our brain works. So for those that have a background in biology or medicine might already know this, but for the rest of us, please bear with me. So we'll go through the basics a little bit. So this is a neuron. You have many of them in your brain and all around your body. And these cells have a particular function in the body which allows it to transfer electrical signals from one neuron to the next neuron. And in that way, you're able to think, you're able to move your body, you're able to experience the world around you. So they're quite important cells in your body to make you, you. So these cells consist of a cell body with a cell nucleus in the middle, and they have a wide network of dendrites where they receive electrical signals, and most neurons have one axon that is most often quite much longer than the dendrites. and at the end, there are a network of terminals where it connects to other neurons to send its signal, electrical signal to. So in this way, with these connections that these neurons make, they're able to connect to each other and hence send electrical signals, receive electrical signals, and send them out again. So we'll now zoom in a level and focus on the cell membrane. So the cell membrane, and this is not specific to neurons, but most of our animal cells have a lipid bilayer around the cell, which means that at one side is the inside of the cell and the other side of the lipid bilayer is the outer world, the rest of your body. And within the lipid bilayer, there is a lot of hydrophobic space. And the thought was, once Meyer and Overton figured out that all these anesthetic agents are hydrophobic, they must have something to do with the lipid bilayer. So over the basically from 1930s till the 1980s, there have been all kinds of theories of how these anesthetic molecules go into the lipid bilayer and create their anesthetic effects. So some of them propose that just by widening the lipid bilayer, it would create its effect. Some propose that the anesthetic molecule would create all kinds of structures around it and creating its effect in that way. So that was the idea of how it affects the lipid bilayer and make it harder for electrical signals to propagate through the neurons in order to have its effect. So in order to understand how electrical signals move through a neuron, we'll have to see how these cells interact. So in the top left corner, you see a neuron in red with the dendrites on the left and the axon on the right. And the green lines on the left are axons from other neurons that terminate in synapses with our red neuron. Most neurons have a stable electrical potential of -70 millivolts, and when spikes arrive from other axons, that level is changed. And that level is changed upwards, but small amounts. And it's only when enough spikes arrive at this cell that at some point all these spikes accumulate, and that is something we can see in the graph, where at some point it reaches the blue threshold and the neuron opens its gates and changes polarity and creates its own spike that travels over the cell and goes to its axon to create spikes on the next neuron, and then it recovers to go back to that level of -70 millivolts. So how does that work? In the lipid bilayer, there are all kinds of channels in it that allow ions to go in and out of the cell. So at a resting potential of -70 millivolts, the cell creates that by having a lot of potassium in the cell and a lot of sodium out of the cell. And that causes the neuron to have that level of -70 millivolts. we focus now mainly on potassium and sodium, but it also involves calcium and chloride, but there are other channels for that. So when those spikes terminate at this neuron, the ion channels open, and and sodium ions are moving into the cell, creating a positive effect on the potential, the cell potential. And till the point where it reaches a certain threshold, and then all the ions open and the signal propagates. The other way around, when after the spike, the depolarization happens, the cell opens its potassium channels, and potassium is going out of the cell, restoring the negative effect. But there's a bit of an overshoot in that, and then after the cell uses a sodium potassium pump, which costs energy to move potassium in and sodium ions out to restore the original situation. So understanding how the cell maintains that resting potential, and how a signal propagates through the neuron, brings us now to a level deeper, is what happens at these synapses. How does these cells interact? And this has been research that has been done mostly from the 80s and into the 90s, and actually research is still done on it, to understand how these synapses influence the normal functioning of our neurons. So you can see here that at the top, this is the axon side of our first neuron, and when a electrical signal arrives in the synapse, the cell releases small vessels containing neurotransmitters that are released in the synapse, and the neuron at the bottom has receptors, the blue mushrooms, that that are specific sensitive to the type of neurotransmitter that is released, and in that way the second neuron is activated. So it's not an electrical connection, it requires neurotransmitters in the synapse to connect with one cell to the other. And the choice of neurotransmitter is important to define what type of connection is made. So there are hundreds of types of different neuroreceptors in the brain, but there are a couple that are specifically important for anesthesia. And as the effects of anesthesia are on the same spectrum as gas narcosis, although quite more potent than gas narcosis in diving, we are focusing on these transmitters for now. And there are two specific transmitters that, two receptors that I would like to focus on. That is what we call the GABA receptor and the NMDA receptor. The GABA receptor is one that once it's opened, it actually releases chloride into the cell. And if we go back to that electrical graph that I showed you earlier, when we release more chloride into the cell, the resting potential that is normally minus 70 millivolts actually is moved down to up to minus 100 millivolts. And you can imagine by doing that, it makes it harder for the receiving cell to actually propagate an electrical signal, because it needs more spikes in order to actually reach the threshold now in order to propagate its electrical signal. So in this way, a GABA receptor inhibits the electrical behavior of the next neuron. The other way around, the NMDA receptor, and the graph shows NMDA and AMPA, but for NMDA, it is specifically calcium that is moving into the cell. So this is an excitatory receptor. So what if this one is opened, calcium moves in and creates a situation where the cell gets a small nudge to continue its electrical signal. So a little bit more into detail. Now we'll zoom in first into the GABA receptor. So the GABA receptor consists of 5 subunits and are in the cell membrane. The cell membrane is depicted in blue here, and it has receptor locations for GABA itself, so the neurotransmitter. So if the neurotransmitter in the synapse arises at this receptor, it opens up, it slightly changes shape, and in that way it allows a channel in the middle for really small chlorine ions to pass and go into the cell to change its polarity. But you can also see that these structures have all kind of other locations where other anesthetic agents can connect to the receptor and change its behavior. And you can here see that there are all kind of anesthetics, including ethanol, which is the normal alcohol that we have in our beverages, is connected to the GABA receptor, and in this way it opens up the chloride channel, and in this way it inhibits our normal neural behavior. And you can imagine if that happens in certain parts of your brain, the normal brain function is altered. So how does that work? Why are certain parts of the brain targeted more than others? And that has to do with the subunits of the GABA receptor. So here you see 5 subunits, and there are up to 18 different subunits currently known. And you can imagine that if there are different subunits in a different orientation in this GABA receptor, that they might have slightly different behavior. And that is something that has been researched over the last 10, 15 years, where we slowly understand that these GABA receptors are available in most neurons around our brain, but the makeup of which subunits are in each of these GABA receptors is highly specific to the location of that GABA receptor in your brain. And that causes that they are specific to the functioning of that brain region to exhibit its specific function. So we'll move to NMDA now as well. So the NMDA receptor is quite similar to a GABA receptor, although its function is quite different. Its receptors are different. So it contains 4 subunits instead of 5, and currently there are 12 different subunits known, but the amount of different orientations is quite limited. It always needs at least 2 of type 1, and then the other 2 can be different, either type 2 or type 3s. So the variety of NMDA receptors is a little bit less. But it works again the same. So if glutamate binds to the NMDA receptor together with glycine, it opens up the channel and calcium is moving into the cell. And in that way, it creates an excitation. We know from ketamine and nitrous oxide that they are altering the effect of NMDA receptor, and we know that it's done by actually moving right in the middle of the receptor and blocking its possibility to let calcium in. So in that way, it changes its normal behavior that the NMDA receptor can create its excitation by basically blocking the entrance of calcium. So we dived into quite a deep, we zoomed into the smallest parts as we currently know them on how all kind of anesthetics and probably also nitrogen and other and nitrous oxide, for instance, have their cause of effect on the neurons. So we focused on lipid solubility and how we first thought over the past century that anesthetic caused their effect by changing the cell membrane. Later on, we learned that it doesn't affect the cell membrane itself, but it does affect the proteins that cause signal transmission between neurons that are actually affected by our anesthetic agents, including nitrogen. And more recently, some people that are actually interested in diving medicine in this field figured out that nitrogen affects the GABA receptor as well. So the lipid solubility is still a thing, but we need to understand that it is just one physical property that anesthetic agents have in order to get to the protein receptor in order to exert their effect. Because over the last decade, and especially around the 80s and the 90s, there were more and more research that showed that certain aspects of the lipid solubility theory didn't work anymore. For instance, certain anesthetic agents based on their lipid solubility were expected to have a great effect of narcosis, but in reality they didn't. And there are certain anesthetic agents that are having two stereoisomers that are basically in structure the same, but are different in their because they're mirrored. Based on the lipid solubility theory, they should have a similar effect, but it was found that only one of the stereoisomers was having an anesthetic effect while the other wasn't. And there are a couple other effects that I won't go into detail where there were more and more evidence that lipid solubility wasn't the only thing. So that's where we can now understand with protein receptors that you need a specific shape in order to fit on the receptor in order to create the effect. And we understand that receptor subunits are the makeup of the different subunits determine which effect is exerted in which part of the brain. So are there any questions so far? And I see one in the chat. So if you have any questions, you can put them in the chat now and I will try to explain them. So the first question is, do the type of subunits make the receptor a GABA receptor? So does a different subunit configuration make a different receptor type, or do they all affect the behavior of the GABA receptor? No, so there are different receptors, but for GABA receptor, which is the receptor that is mostly affected by most anesthetic agents, there have been tons of research about this one receptor, and we know that there are 18 different types of subunits, and in different configurations of subunits, they're all GABA receptors. They are all letting chlorine in, and they're all binding GABA neurotransmitter, but we know that they are sensitive to different types of other anesthetic agents. And we know that different configurations are that certain configurations are specific to certain brain regions. Does that answer your question, Tim? Okay. Any other questions about the small details of how neurons work? I hope it is a bit, you guys were able to understand my train of thought into the smaller parts of how our brain works. Because with this knowledge, we can now zoom out and look at the brain as a whole. So for those of you who haven't seen brains before, this is a cartoon of a brain where it is a transect right through the middle, and we look into one of the sides of the brain. So on the outside, we see the cortex with the different colors, and the little brain is underneath, and the neurons that are leaving to the spinal cord are at the bottom, that move into your body. So we'll focus mainly on the cortex for now, and the cortex is highly specialized in function. So certain functions are specific to certain locations on the cortex. So in the front, on the left side, we have the blue part, which is the prefrontal cortex, which is specific to all kind of functions that are related to decision making, to higher cognitive thinking, mathematics. All these type of functions are in the prefrontal cortex. And in the middle, the purple part is the cingulate cortex. And the cingulate cortex is mostly known for connecting all the different brain regions and making sure that information travels from one part via substations to other parts of the brain, to make sure that mostly the prefrontal cortex is aware of all the sensory information coming in. But you can imagine that the prefrontal cortex can't receive all information, because then it will be overloaded. So the cingulate cortex is also related to making decisions on which information is moved forward. The last part I want to focus on is the occipital lobe at the back, the orange part, which is highly specific to your visual functioning. So your eyes are connected to that, and it has the basic functions of recognition of what you see, and the higher functions of making sense of if that is important or not, and if that information needs to be relayed to the rest of the brain to make decisions. So now we move to the, So the cortex is connected to the brainstem, which you can see in the middle at the bottom, which is connected to the neurons that move out of the body. And the brainstem has all kind of small nodes that are highly specific in function, that's our basic brain. So the cortex is quite specific to animals that are having all kind of higher cognitive functioning, mostly mammals. And if we look at evolution, the more simpler the animal, the smaller its brain, and at some point it doesn't have a cortex anymore. It has only the little brain and the brain stem. And if you go to really simple animals, it doesn't have this little brain anymore. It only has a brain stem. And in the brain stem, for instance, there are nodes that regulate your breathing, that regulate your blood pressure, all the really basic functions to make sure you as a animal survive. But there are also nodes in there that connect to the cortex to make sure that basic functions like sleep, like emotions, are sent up to the cortex. And this is a highly simplified network of how these nodes at the bottom are connected to the cortex in brown at the top. And you can see that each of these nodes have different neurotransmitters. So for instance, GABA in red, orexin in purple, well, forgot about that one, in green, and they have their influences on the cortex. But more importantly, propofol, a highly used anesthetic, which is a GABA antagonist, affects both the cortex but also these lower brain nodes, and inhibits their function, and hence it causes narcosis. And we're not entirely sure, and that's still area of research, where propofol mainly attacks the cortex, and because the cortex goes silent, it creates narcosis, or if propofol attacks the basal neural nodes and silence them, and hence creates silence in the cortex. That's two hypotheses that haven't been proven yet, either a top-down or a bottom-up approach. But we do know that these agents are attacking multiple locations in the brain to create their anesthetic effect. And similar does nitrogen. We know that nitrogen affects some of these neural nodes in the basal ganglia, but we also know that it does attack some of the higher locations in the cortex directly. And you can imagine that if it attacks specifically certain receptor subunits that are specific to the frontal lobe of your cortex, that it has specific function deficits like decision making, like memory. So my research has been in my research I've used EEG, so measuring the electrical signals of the brain in order to measure how gas narcosis works on neurons. But in order to measure these electrical signals on the outside of your skull, we need not the effects on one neuron, because those electrical signals will be way too small to measure, but we need effects that are on brain regions that happen synchronously. So luckily, the effects of narcosis are affecting multiple neurons at the same time, and hence these effects can be measured on the outside. So the changes in neuron excitability create differences in the electrical fields that are created next to the axons, and these electrical fields travel through all the layers of tissues that are between your brain tissue and the outside world, including your skull. And you can imagine that you need quite a strong electrical signal in order to be able to measure that. So my first study that I did, before I actually start, are there any more questions, or is this all clear? Okay, so the first study I did as part of my research into gas narcosis was using nitrous oxide, which some of you might be familiar with under the more common name of laughing gas, which is a recreational drug used as well. And I chose this anesthetic drug specifically because back in the 80s, nitrous oxide was used quite commonly as a substitute of nitrogen narcosis, because it is a more potent gas than nitrogen, And hence, we don't need a hyperbaric chamber to do experiments. We can just do them in the lab by having people breathe a certain percentage of nitrous oxide. And these studies found that the behavioral effects of nitrous oxide are quite similar to the effects of air narcosis at death. And most studies found, or a couple of those studies found, that 50 meters of diving has an equivalent effect of around 30 to 35% of nitrous oxide. Okay, cool to hear. So in the first research project that I chose, I wanted to use nitrous oxide as well, because it would allow me to do longer experiments and having multiple exposures without having to deal with the environment of a hyperbaric chamber. And the two main drawbacks of using a hyperbaric chamber is, one, if you do experiments at 50 meters, your time is fairly limited, because every minute you spend there is 2 minutes of decompression. So you can easily see doing an experiment for an hour or two means you're basically sitting in a hyperbaric chamber for a whole day, which is quite hard. And the second part is that the hyperbaric chamber is quite restrictive on what type of electronics you can use to measure the effects of nitrous oxide, of narcosis, because of fire risks with using electronics inside the hyperbaric chamber and the pressure effects on the electronics itself that can damage the electronics. So that's why we set off with a simpler experiment first, using nitrous oxide and figuring out which tests I want to use specifically, and figure out how much time I actually need, and try to minimize figuring out which parts are important for my study before I would commence my study inside the hyperbaric chamber. So Tim, I'll come to that in a second. So I set off in this experiment where we measured EEG, as I just explained, and all kind of other physiological effects of narcosis, including pupillometry, the size of your pupil, and psychometric testing. But I won't go into those in this presentation. So the head plots you see in the figure are the exposures of baseline. So breathing, in this case, 50% oxygen and nitrogen, and then 20% of nitrous oxide in oxygen, 30%, 40%, and a final measurement at the end where the participants were breathing air again. And there was every time a rest period in between where participants were breathing normal air again to recover from the effects they just experienced. And this is not a simple electrical activity of neurons, because we tried that, and the effects of nitrous oxide are way too subtle to pick that up in that measurement. So we created developed a algorithm to analyze the EEG. And for those that are interested in it, I can later on explain the different analysis steps that are involved to create a head plot like this. But it basically comes down to how complex the electrical signal is by using recurrence analysis and the Shannon entropy. For those nerds that are more towards signal analysis. So what we see here is that at baseline, the middle of your brain has quite complex signals, which means that over time, the signal changes quite a lot. There's not that much recurrence of signal happening. And once we administer nitrous oxide, we see that that yellow blob is getting smaller and reduces its complexity. And that's where we built a metric on what we called the default mode or DMN, default mode network, DMN complexity, because this middle part of the brain is thought to overlap with the default mode network. And that default mode network is in the cingulate cortex, the purple area that, as you might remember, I explained earlier, is connecting the different parts of the brain. So our thought is that nitrous oxide creates an environment where the default mode network is not able to have complex information sent around the brain and hence disrupts information flow between brain regions. So with this metric, which took quite a while to develop and optimize, we had a powerful analysis method that is highly sensitive to the effects of nitrous oxide, which would be great to now use on our next experiment, where we have divers go into the hyperbaric chamber and experiencing nitrogen narcosis. So that was the next study we did. We had, again, 12 divers come into our laboratory, and this time they came into the hyperbaric chamber, where we exposed them to 6 atmospheres, equivalent to 50 meters. And one time they came in, they breathed air at depth, and the other time they came in, they breathed heliox, which is a mixture of 80% helium and 20% oxygen. So in this way, we will be able to measure the differences between nitrogen and helium. And helium is a gas that is believed to have no narcotic effects within these depths. So in that way, we were able to see if there is a specific depth effect or a effect of nitrogen itself. That's Stefan, you're asking, what about that increase of complexity after? That's a really interesting question. And yes, we noticed that as well. We're not quite sure what that means. There are some ideas, and especially with ketamine, which is a NMDA receptor as well as nitrous oxide, that it has some longer lasting effects of being able to have the brain being what we call more able to reconnect with it between brain regions. So it might be that that is what we're measuring, but that
like an increase in sensitivity also.
Sorry?
Like an increase in sensitivity.
Yes, something along those lines. Yeah.
Yeah. Okay. Interesting.
Yes, it is definitely interesting. And this is a paper that we recently published in anesthesiology, because there's not that much known about the nitrous oxide and ketamine, and there's more and more interest in these agents, and especially also in especially ketamine in well as an anesthetic, but also as a drug to alleviate some of these symptoms of depression because of its long-lasting effects of allowing the brain to what we call rewire. So allowing to form new connections, and in that way probably has some effect on the symptoms of depression. But that is an active field of research currently. And nitrous oxide might have similar effects although probably to a lesser extent because it's less potent than ketamine. So,
great, thanks.
Sorry?
I said great, thanks for the explanation.
So back to the hyperbaric experiments that we did. So we had 12 divers going into the hyperbaric chamber twice, breathing once air, once heliox. And we thought, okay, let's use our newly developed algorithm to hopefully be able to record and analyze these EEG signals again and see if we can measure the same effect. So we did all these experiments, we analyzed the EEG using the DMN complexity, and these are the results. Quite unfortunate, nitrogen doesn't have any effect on DMN complexity. So there goes our idea that nitrogen has a similar effect as nitrous oxide. And one of the ideas that we developed to explain this is that although both gases have a similar behavioral effect, we know that nitrous oxide has an effect on the NMDA receptor, while nitrogen probably has an effect on the GABA receptor. Their way of how they change the behavioral effects on a neuronal level might be different, and hence we might not be able to pick up on that effect using our newly developed DMN complexity. So the last year I've spent more time analyzing EEG because there are quite a lot of different ways how you can analyze the EEG. And one of the methods that I've been working on recently is what we call functional connectivity. So in this analysis method, we're not looking over the recurrence over time and the complexity changes over time, but this time we're looking at how similar are signals between electrodes. And luckily, we found that one of the functional connectivity metrics called mutual information is able to capture the effects of nitrogen narcosis. So what we see here, that at baseline, there are a couple of connections, which means that the signals between these electrodes are quite common. But if we increase the nitrogen pressure by increasing the pressure, so the second one is air breathing at 2.8 atmospheres, being 18 meters, or the third one breathing air at 6 atmospheres, 50 meters, we can see that the signal becomes more common between electrodes. And this is thought that this is caused by inhibition of the neural activity on the cortex itself, and hence these areas of the cortex all start to have the same signal that comes from the thalamus, which is a brain region that is one of those brain nodes deep inside the brain that is highly connected up to the cortex. And if the brain is not doing it, these brain regions are not doing their own thing, they kind of go to a basic idle rhythm, which is dictated by the thalamus. And that means that all these brain regions are having more of the same signal, and hence the same information in those signals, which is picked up by our mutual information analysis. So that is what I've been doing over the last, well, probably year, is analyzing EEG and figuring out these things. And these are the results that we've come so far. So you must understand that if you look at the EEG signals themselves, which are 32 of scribbly lines, you won't see with the naked eye that these are similar. It's not like they're all a nice wave doing the same thing. But the subtle changes that nitrogen caused can be picked up by this analysis that is sensitive to these small changes that are picked up. So with that, I would like to conclude that in the studies that I've done so far, is that our metric of DMN complexity is sensitive to the effects of nitrous oxide, which is an NMDA agent, while functional connectivity is sensitive to the GABA effects of nitrogen. And we must understand that the complex of consciousness is quite likely not a linear line of where you're traveling on, but a multi-dimensional space of where there's only a small hotspot of where we are currently functioning at that allows our brain to function the way it does. And if you alter it in small different ways, it can kind of fall off this mountain, this hot zone, and fall into a great lake around it that in the end causes unconsciousness. But and on that slope, there are different levels of altered consciousness. So that was my presentation about gas narcosis. I hope it helps you to better understand that gas narcosis is quite a complex effect on the brain. Hopefully in the future, we will be able to measure this effect continuously real time, and in that way create a monitor, probably mostly for commercial and military divers, to monitor their brain state, and in that way make their diving a bit safer, because you can imagine that these divers are doing quite complex dive, diffusing mines, all these kind of quite dangerous jobs, and we need to make sure that they're capable of doing that safely. So Stefan, your question is, so some sort of oscillations of signals causing higher recurrence, is it then incapacitating synapse functions like increasing capacitance?
I'll try my best.
Yeah, thank you. It's quite hard to link a single neuronal change because we understand the effects of narcosis quite well, reasonably well on a cellular level. We understand the effects of narcosis on a whole animal level, but the steps in between of how it interacts with networks and how it exerts specific changes in behavior is something we are not fully understanding yet. So to answer your first question, so we kind of have an idea now that that nitrous oxide is indeed creating a higher recurrence of signal, and hence a less complex signal over time, where nitrogen affects the neuronal effects, where the complexity decreases, but complexity in a spatial way. So not over time, but over space. And how that specifically work on a synapse level and how it increases capacitance, I'm not entirely sure.
It's just my sort of trying to get my brain around it.
Yeah.
You said capacitance.
Yeah. So there are electrical models of cellular functioning. One of the most basic ones is what we call the Hodgkin and Huxley model, which models the electrical signals, the electrical functioning of a cell, and then you can change certain parameters of each of the ion channels that are in that model that changes the electrical behavior. And these models are quite commonly used to model neuronal behavior. And with increasing computational power, they're now capable of modeling up to 1,000 to 10,000 neurons and create networks of them to mimic the effects on a network level. So with increasing computational power, we slowly start to understand how networks work. But the problem is that computational power is not strong enough yet to model the millions of neurons that we have in our brain. And we don't have enough knowledge yet to distinguish how all of these neurons are slightly different and how their behavior is slightly different. So yes, it is a puzzle that slowly is unraveled of how our brain works. And over time, we'll get to know our brain a little bit better every step.
Then we can inject memories.
Sorry?
And then we can inject memories, right?
Elon Musk with his Neuralink is quite an interesting project he is doing to connect to small patches of neurons, but in a highly dense way that he's able to measure individual neuron activity, but he can also send electrical signals back into the brain to alter their behavior. So it's an interesting project, and we'll see what comes out of that and how that can advance our understanding of how the brain works. So Fokke, you have a question about, so in the future measuring EEG underwater. Well, there is already one research group that has shown to be able to measure EEG underwater in a pool at 5 meters of depth using an EEG system with a bathing cap over it to keep it dry, because you can imagine measuring really small electrical signals in a highly water environment is challenging on its own. For our project, we currently see that it is more towards military and commercial diving, because they use helmets that are reasonably dry. So in that way, it becomes easier to measure EEG in that environment with something like a sticker or a bathing cap to keep it really dry.
And astronauts as well?
Sorry?
And astronauts as well?
Who knows? Maybe. I don't see yet how their why we would want to monitor them other than out of scientific interest, because they are in a environment that is having a normal amount of oxygen and no narcotic agents. Their pressure is a bit lower than we have on Earth, but overall, I don't expect to see major EEG changes. But who knows? Maybe at some point, I would be opposed to that, to work with them.
And they also practice in large swimming pools.
Yes, they do.
I don't know how deep they are, but
I'm not entirely sure, but probably not too deep, because they're not interested in depth itself. They're more interested in the weightlessness environment that the pool creates.
I believe 20 meters of depth or something like that.
So the next question is gas narcosis something you can get used to? So as I alluded from at the start of my presentation, you can get used to the effects of narcosis. It doesn't mean that the effects of narcosis go away. It just means that you're able to cope with them better. So you know how they affect your body and how you can compensate for that in order to still achieve the task you're intending to do. And it's the same with alcohol. If you're a regular drinker, you're able to walk better in a straight line than somebody who hasn't been drinking for quite a while and then drinks the same amount as you do. But it doesn't mean that the symptoms are not there. You're just better able to cope with them as long as you're doing tasks that you know how to do. And that's the problem with gas narcosis in a diving environment, is that sometimes you will encounter environments where you have to make decisions and do skills that you're not familiar with. And especially a rescue or situation can be quite challenging on its own without narcosis. And you can see why narcosis is influencing rescues, and we see more fatalities and serious incidents at depth than in the shallows, because people are less capable of solving them to a good result. Cool. Any other questions? Would the fact that your test subjects are divers influence your test? Good question. I don't know. I haven't tested naive divers. And one of the reasons we chose to measure divers is because it makes it easier to find participants, because they're having an interest in this research. And as Stefan already mentioned, he would be happy to apply for a trip to New Zealand to participate in the study. So are divers here. They're quite keen on participating in these studies because they're quite interested in diving medicine, and being in a hyperbaric chamber is already quite an experience for them as well, because most divers have read and heard about hyperbaric chambers, but most divers haven't seen one, let alone be in them to experience the effects of gas narcosis at 50 meters. So that's why we choose divers. And the other part is that there are some risks involved in going in a hyperbaric chamber, similar to diving. There is a small chance on decompression sickness. There's a small chance of getting oxygen toxicity. And it's way easier for us to explain these risks to divers than it is to explain that to naive laypersons. So that's why we chose divers. It could well be that the EEG of non-divers would be different. I don't know. But it is more relevant to our research and our overall aim to have divers, because that's where we, in the end, we hope to see this project going, is that we might be able to create something that is a continuous monitoring method for divers. So how does that relate to hypoxia? That's a whole other research avenue, where there is some research, some papers that show the effects of hypoxia on EEG. But you must realize that although the recording of EEG has been around from the 60s, the advancement of electronics and especially computers have only allowed the analysis of EEG more than just frequency band analysis with Fourier transforms, which has happened, well, basically since the 80s. Most of the research in these small fields are only have been done sparsely, and hence have been done in those periods where only the simple analysis of frequency band analysis has been done. And that only captures really broad, powerful changes in order to be captured by these analyses. And it's only in the last, let's say, 10 years, 10, 15 years, where the field of neuroscience and especially EEG analysis has taken off tremendously with the amount of analysis options that we have nowadays, that changes our ideas of and understanding of how these physiological changes affect our brain. So actually, we are currently applying for a grant to continue this line of research on gas narcosis, but also to extend it to a group where we will expose them to hypercapnia and hypoxia, and measure EEG as well, and see how, if we can pick that up in the EEG, and if we can create, if the same metrics are sensitive to that, or if we can develop a different metric that is sensitive specifically to hypoxia. Both would be interesting. The next question from Dan. You mentioned that how nitrogen narcosis is taught and explains is outdated, not accurate. What would you explain differently as an open water instructor? Good question, and I can see why you want to know that. I think especially, some of the materials, especially if you move into more technical diving, explain that where we talk about oxygen narcosis and the effects of CO2, you quite often see the Meyer-Overton rule and tables of lipid solubility factors between nitrogen and other gases, and saying that because of their level of narcosis, level of solubility, we expect CO2, for instance, to be 20 to 25 times as narcotic as nitrogen, or oxygen twice as narcotic as nitrogen. And I would be reluctant to use that information to tell divers if certain gases are narcotic, just based on their lipid solubility. One of the studies that I haven't shown the results of, because we're still working on that, but we also did a study where we exposed divers to oxygen in the hyperbaric chamber, where we measured their EEG at the surface with 100% oxygen at 4 meters, which is 1.4, which is a quite common maximum limit of oxygen exposure in normal recreational diving, and also to 2.8 atmospheres of oxygen, which is the limit we can do in the hyperbaric chamber safely. And we saw that we can measure that effect as well, but quite differently than nitrogen. So we're working towards that to probably come to the conclusion that oxygen is not narcotic at all, at least not in the same way that nitrogen is. So that is one part where I would be reluctant as an open water instructor to, or as a diving instructor, to tell my students. The other part is and that comes more down to the physiology chapter in, for instance, Science of Diving in the SSI materials, and Paddy has something similar in their encyclopedia, is quite a detailed explanation of Meyer-Overton, of the cell membrane theory, and some other theories from those 50s and 60s. And that's where they stop to explain what gas narcosis is. And well, if you want to go down that route of explaining how gas narcosis work, you need to tell the whole story and not stop basically 50 years ago. So that's the other part as a diving instructor, where I would change the narrative of that story, which is something I will probably do once I've finished up my thesis, is come in contact with both Paddy, SSI, and probably also some other diving organizations, and start spreading basically this story of explaining this, because not only divers only know that's this first small bit, but even diving medical physicians are quite unaware of this second part. And that's a shame, because these are the people that hopefully tell the right information to other divers when they're out about in the diving community. Cool. Any other questions? Okay. You can
So
also,
how long to go before your PhD is done, Xavier? Are you writing your thesis currently, or?
Yes. So I've, although I every time say I'm done with the analysis of my data and I'm currently in the writing phase, when you keep on writing, and you probably know yourself as well, you come back to the analysis to change small things, or do another analysis to make your results more clear. So I'm in that phase currently, where I have 3 publications done. They are published, and I'm currently writing the nitrogen narcosis chapter and the oxygen narcosis chapter. Those are on their pace, so the first draft is done, and the nitrogen narcosis chapter is in the review, has been with my supervisors now a bit. So we're working towards finishing those. And then I have to write a small part of my literature review on EEG analysis still, and my general conclusions, and then my thesis is done. So probably a month or 2.
Ooh.
Yeah, so it's getting pretty close.
Yeah, it is.
So the next question is, maybe you explained it at the beginning as well, but as a student, I was curious to know whether being used to the effects of alcohol would help you fight the effects of nitrogen narcosis. Yeah, that's an interesting question, and I am not sure if there's research out there that has done that. No, I don't know. So I can't answer that question. Sorry. Probably we don't know, and also because the effects of nitrogen narcosis do vary, it's quite hard to do such research because the effects will be quite small. I'm
very curious why you're asking, but
and Dan asked a question. There was an article last year or so about the effects of nitrogen narcosis did not completely disappear when ascending. It was something you also included in your research. The project looked at pupil responsiveness, I think. Yes, I think you're referring to one of the Den publications that was out there last year, I think. You might refer to the, well, I don't know which one, but that draws back to a paper that has been published probably 10 years ago, where they used indeed not pupilometry, but what we call the critical flicker fusion frequency, or short for CFFF, which is a measure that measures your sensitivity to flickering light and your ability to see it flickering. And if you increase the frequency enough, at some point you won't perceive it flickering anymore. Yes, it's the FIPO project one. Yeah, I know that study. And CFFF is a method that we used in our research here as well. And that's mainly due to when I designed this study, I was not willing to put all my eggs into one basket into the EEG side of things, because I was well aware that I might not be able to find any changes in EEG. And well, writing a whole thesis with only negative results is a challenge on its own. So that's why when I designed these studies, I included CFFF as well, because I know that there are around 10, probably around 10, 15 studies from a group in Belgium that has used CFFF extensively and was able to measure narcosis already at 30 meters with it. So my idea was to use CFFF as well. So something I didn't present today, but that is one of the papers we published where we used CFFF, including one of their devices, inside the hyperbaric chamber at 50 meters, and we didn't find any change in CFFF. We repeated this study a second time at 50 meters using CFFF with a different device, and again, we didn't find any changes. So I know that this group finds all kinds of effects with CFFF in multiple environments and publishing them in all kinds of journals. But having my own experience using CFFF and having been in communication with the professor that uses that CFFF in Belgium extensively, and he was part of that study here as well. I would be reluctant to completely draw conclusions from experiments that have been done with that that method. So yes, I know that study is there, and I know that that study showed a impairment 30 minutes after diving on using CFFF. But I'm not entirely sure what they're actually measuring, because diving is also quite a fatiguing exercise. You're consuming quite a lot of energy. So you could also argue that they're just measuring fatigue at the end of the dive and not nitrogen narcosis. So that is something we don't know. But I would be reluctant with that, with those studies to draw conclusions from. Any other questions? Can be also broader about diving physiology, or yeah. And you can ask them over voice as well. You don't have to type if you don't want to. The group is getting smaller, so